that introduction. That's okay. I can just. Yeah. Okay. So my name is Anna. I've been working in the Egler lab, and this is the title of my project. It's a little bit dense, so I'll walk you through it. But basically, we, as the Egler lab, are studying a protein called NRF2, which controls antioxidant response. And we're really interested in how NRF2 functions within our body. So, the treatment of chronic diseases, specifically cancer, heart disease, diabetes, and neurodegenerative diseases, accounts for almost 90% of healthcare spending in the United States. How can we address this? One exciting avenue is through the NRF2 protein. And that is because the NRF2 protein and its activation is implicated in mitigating or preventing altogether all of these illnesses. Even more interesting is that disease prevention provided by NRF2 can actually be found in the diet. Specifically, the NRF2 protein is activated by compounds that are naturally found in our foods. For instance, resveratrol is found in the grapes that are used to make red wine, and it activates NRF2. EGCG is also an NRF2 activator, and it's found in green tea. Sulforaphane, another activator, is found in cruciferous vegetables such as broccoli sprouts, and I'll be talking a lot about sulforaphane today. Notably, all of these compounds are currently in clinical trials to evaluate their effectivity at treating illness in humans. How do we know that these compounds activate NRF2? Well, researchers have generated an NRF2 knockout mouse, which is a mouse model that's lacking the NRF2 protein. And these mice are sensitized to many different detrimental conditions within the body. For instance, point with my hand, these mice are sensitized to skin and stomach cancer. Typically, compounds found naturally in foods shown in these red boxes are effective at diminishing these negative effects. But in NRF2 knockout mice, this is not the case. For instance, sulforaphane usually can treat skin and stomach cancer, but in NRF2 knockout mice, this ability is significantly diminished. This indicates that these activators work through NRF2. So I've been talking a lot about NRF2, but what we're really interested in is how it actually works. So to do that, I have to explain just one term, and that's electrophiles. So electrophilic means electron-loving, and these molecules are drawn to places in our body that are electron deficient, and in this way, they're able to respond to different conditions. So normally, the NRF2 protein is bound by a repressor protein called KEAP1, and this targets it for degradation in the molecular trash can of the cell. However, when cells are exposed to electrophiles, such as the compounds that I mentioned on the first slide, like sulforaphane found in broccoli sprouts, NRF2 is no longer bound by KEAP1, and it can accumulate within the cell. Once it accumulates, it can bind to a series of genes called antioxidant response elements. And when turned on, these elements are capable of producing proteins such as antioxidant and detoxification proteins. And one of these proteins is AKR1C1, and I'll be talking about that later in my talk. So, it's been well established that electrophiles upregulate NRF2. However, something that's complicating this question is that many of the electrophiles also produce something called reactive oxygen species. You may have heard of reactive oxygen species or free radicals, and perhaps you've associated them with something negative, but they're also really important as signaling molecules within our cells. So TBHQ is an electrophile, but it also produces a reactive oxygen species called superoxide through redox cycling. But the role of these reactive oxygen species in contributing to AA reactivation is not very clear. Some researchers have published results that show that reactive oxygen species are not really relevant to AA reactivation. But other researchers have published results that clearly show a ROS dependence in AA reactivation. So this discrepancy within the field is something that we really wanted to get to the bottom of to help to understand how NRF2 protein works and how we can best take advantage of it for health. So for that reason, our main goal is to assess the role that reactive oxygen species generated by NRF2 redox cycling activators plays in NRF2 AA reactivation. And our hypothesis is that reactive oxygen species activate ARE minimally on their own, but significantly enhance ARE activation in the presence of an electrophile. So to address this hypothesis, I've been studying Hackett cells, which are human, non-cancerous skin cells. And I've been treating them with a variety of treatments. Treat them with sulforaphane, which, as I mentioned, is an electrophile found in cruciferous vegetables. This is the picture of sulforaphane, and in red is its electrophilic portion. I also treat with reactive oxygen species, sometimes in the form of hydrogen peroxide that we buy in a bottle, like this, which is something that you might use to treat a cut when it needs to be disinfected. I also treat in the form of reactive oxygen species that have been produced through redox cycling. So here is DTBHQ, which is similar to TBHQ, which I showed on a previous slide. But DTBHQ is not an electrophile. So its only purpose in this assay is to produce reactive oxygen species through redox cycling. And then finally, I use catalase, which quenches hydrogen peroxide. So this is one of our results. So it's called a Western blot, and it shows protein accumulation. So these bands indicate protein accumulation within different samples, and specifically this is of AKR1C1, which, as I mentioned, is a detoxifying protein that's controlled by NRF2 and ARE. So to interpret this Western blot, we look at the darkness of the bands. So in lanes 1 and 2, when it's just white, that means that there's minimal or no protein produced. So we treated cells with sulforaphane and DTBHQ. And in lane 3, you can see the AKR1C1 accumulation in cells treated with sulforaphane. And then we compare this to lane 5, which is of a sample treated with sulforaphane and 5 micromolar DTBHQ. And you can see that the band becomes darker, indicating that more AKR1C1 is produced in the presence of reactive oxygen species. So this supports our hypothesis that reactive oxygen species, in combination with an electrophile, such as sulforaphane, activates ARE. And then if we look at lane 7, this is just a higher concentration of DTBHQ, we see the band become even darker, which again confirms our hypothesis. So this is a pretty typical result that we've seen quite a bit. However, sometimes when we repeat the assay, our results become a little bit different. So I'll show you that. So this is another Western blot, ostensibly conducted in the same way. Again, we have cells treated with sulforaphane and DTBHQ. However, this time, when we add 5 micromolar DTBHQ to cells treated with sulforaphane, we do not see an increase in AKR1C1 expression. 5 micromolar DTBHQ, in this case, was not sufficient to increase ARE and NRF2 activation. we need 15 micromolar DTBHQ to see a similar response. This was kind of frustrating for us because we were confident in our results because we had seen them a lot. But every so often we would get this anomalous result, and we wanted to publish, but we didn't feel like that would be appropriate. So we started brainstorming about possible reasons for these discrepancies. We thought of a lot of things, but we narrowed it down to cell conditions. So I work with Hackett cells, and there's 3 things that we hypothesized could be affecting the cells. One, cells grow in a plate, and sometimes they're closer together, and sometimes they're farther apart, and this is called cell confluency. So we thought that might be affecting the cells. Cells are grown in media, which is like their home and their food, and we thought that perhaps the age of the media could be affecting the results. Finally, we considered the possibility that when we changed the media before treatment, which is something that we did kind of not super regularly, that could also contribute to variability. So I'll go into those 3 things in detail, but just to give you a little overview. So our goals were to identify the causes of variability in results, publish our key findings, and better understand the mechanism of our treatments within our cells and how cell maintenance conditions affect our results. So I showed you a Western blot, and I just kind of interpreted the darkness of the bands. However, the Journal of Biochemistry has recently enacted new author guidelines with stricter suggestions for how to report Western blots. Specifically, they noted that sometimes it can be pretty subjective to just decide which band is darker and which is lighter, and you definitely can't say by how much something is darker. However, we can use a computer software to quantify the bands into numbers, which we can graph, and then we can confidently report our Western blots. To do this, however, we needed to make sure that the signals of our proteins when we graph band intensity versus sample concentration had a linear correlation. So I have here 2 titrations of sample concentrations of cells, and I probed by fluorescence for either beta-tubulin, which is our loading control, or for AKR1C1, which, as I mentioned, is our protein of interest, a detoxifying protein controlled by NRF2. And as you can see, when we graphed band intensity versus sample concentration for both of these proteins, we saw a linear correlation with high R² values and a 2-fold increase in sample concentration, approximately correlating with a 2-fold increase in band intensity. And that was really positive because it meant we could begin to quantify our bands and confidently report our Western blot results. Another way that we've been measuring ARE activation is via a luciferase assay. So in this experiment, we put next to the ARE gene a gene for luciferase protein. And this is a protein that, when turned on, produces luciferase, which makes fireflies glow. So when NRF2 binds, it turns on ARE, and it also turns on this luciferase gene. We can measure the amount of light produced as a proxy for NRF2 activation. So I'm going to show you some assays that use light as the readout. So this is one of the assays, and it was to test whether changing the media before treatment increases ARE activation. So the reason why we narrowed this down as a possible source of variability is because, well, we didn't actually at first. So what happened was that we had been changing the media sometimes before experiments, and we weren't really keeping it consistent. Sometimes we were changing the media 2 hours before treatment, and sometimes 1 hour, and sometimes we weren't, related to other things, but we didn't really think it would affect our results. However, after we did a little literature search, we found a not super well-known fact to us that some cell lines actually secrete catalase into the media. And as I mentioned, catalase quenches hydrogen peroxide into water and oxygen. And you could imagine if that cells are secreting catalase and the very same cells are being treated with hydrogen peroxide, that perhaps the catalase was quenching the hydrogen peroxide that we were trying to treat the cells with. And this would, A, diminish A reactivation, and B, be a potential source of variability. So to test this, we first conducted an ARE assay with cells treated with hydrogen peroxide with no media change. So this is a nice little picture of how the cells grow on the bottom of the plate, and then there's media on top. So this is a typical assay where hydrogen peroxide on its own minimally enhances AR reactivation, but in combination with an electrophile, namely sulforaphane, we see a greater activation. When we changed the media before treatment, we saw a significant increase in AR reactivation. So this indicated two things to us. A, that it did appear that our cells were secreting catalase into the media, and that this was actually diminishing the AR reactivation that we had been measuring. And B, that this was a potential source of variability that we could easily control by changing the media beforehand each and every time. So we're pretty excited about this. So another source of variability that I had mentioned is cell confluency. So when cells grow on a plate, they cover the plate to varying degrees. And when we first put them on the plate, there's just a few cells, and that's called low confluency. But then they grow, and then they cover more of the plate. So the ideal time to take cells from one plate to another, as well as to treat them, is at 75% confluent, meaning that cells cover 75% of the plate. And this is an actual picture I took of my cells, when they were 75% confluent. So it seems pretty simple, but it's actually difficult to keep the confluency consistent. The cells don't always grow at the same rate, and when we're bouncing between classes, it can be hard to catch them at the right time to treat them. And then another reason why confluency can be variable is because it's a little bit difficult to determine exactly what the confluency is. Like, how do we know this is 75%, not 70 or 80%? So, but we really wanted to get to the bottom of the effect of cell confluency. So we plated cells at 4 different confluencies, C1 being the lowest confluency, and C4 being the highest confluency. And for each confluency, we didn't change the media beforehand, and we did change the media beforehand. So let's look at C1. As I showed you in the previous slide, when we changed the media, and these cells are treated with 2 concentrations of hydrogen peroxide. So when we change the media before treatment, we see a significant increase in AR reactivation, once again confirming that catalase is most likely being secreted by the cells and affecting our treatment. So that's good. However, we were really convinced that we would see differences between our cell confluencies. And when you look at this, for instance, if you look at C1 media change versus C2 media change, these values are not statistically different. All the confluencies at one media change show basically the same AR reactivation. So we were really stumped by this because we had seen previously that cell confluency was definitely affecting our results. But when we actually did an experiment to test it, we weren't seeing any difference. So there was just one more variable that we thought might be influencing it, and we tested that next. So that was age of the media. So the media comes in a bottle, and then we open it and add a few things, and then we put them in our cells. And sometimes the media is sitting in the fridge for a week or so, and sometimes we open a new bottle and we use it right away. So we definitely had not been keeping that consistent. So this experiment shows the results of when we use fresh media versus old media, and we have cells plated at 2 confluences. The light colored bands are low confluences, and the dark colored bands are high confluences. So let's look at cells using old media first. So this part is low confluency and this part is high confluency. When we look at these cells, we can see that there's a little bit of a difference in AR reactivation in cells treated with hydrogen peroxide versus low confluency and high confluency, but not a huge difference, not really as much as we had been expecting. However, when we look over to in the new media, there's a huge difference between, let's look at this green bar at low confluency and this green bar at high confluency. So this indicates that actually ARE response is multi-variable dependent. So the confluency affects the ARE activation more when using fresh media than when using old media. So we were actually really excited to get these results because we finally pinned down why sometimes we had been seeing an effect of confluency and sometimes we hadn't been. So to conclude, we have identified the major sources of variability. Namely, cell confluency affects ARE activation mostly when using fresh media. And we're really excited to repeat our experiments using this new knowledge because we think that we can confidently support our hypothesis. And our preliminary data indeed supports our hypothesis that reactive oxygen species significantly enhance ARE activation in the presence of an electrophile. And perhaps even most exciting is that because we've identified this information about reactive oxygen species, this can perhaps be used in the future. For instance, we can use reactive oxygen species to make broccoli sprouts more healthy. What I mean by that is that reactive oxygen species activate cells treated with an electrophile, which means that perhaps we could support treating with sulforaphane, which is already in clinical trials, with low amounts of compounds that produce reactive oxygen species. And maybe because reactive oxygen species increases NRF2 and ARE activation, we would need to use less sulforaphane or other electrophilic compounds. And this is cool because high concentrations of any compound can be cytotoxic, which we don't really like. Yeah, and you could imagine, for instance, supplementing sulforaphane from broccoli sprouts with a compound that produces reactive oxygen species, like EGCG from green tea. I have a lot of acknowledgements. So first, I'd like to thank the Falvey Memorial Library for resources. And so obviously, working in a basic research lab, a lot of the data comes from things we've generated, but the resources from the library really play an important role in supporting our data and putting it into context so that we can know what researchers across the country and around the world are doing as well. We have, I think, over 100 references saved for this project alone. So the databases that we have access to via the library have really been crucial in supporting that. I would like to thank my mentor, Dr. Amy Egler, who, as she mentioned, she took me in as a freshman when I didn't really know what I was doing, and she supported me and gave me independence on projects and really taught me what research was and cultivated my love for it, so thank you. I would like to thank my Egler lab mates for their help and friendship. So Brad and Nick graduated. Nick was a former Falvey scholar as well, and they helped me when I was a freshman who didn't know anything and didn't know what I was doing. And Linda, who's my fellow senior, we've been doing a lot of these experiments side by side. So sometimes when I need to bounce to class, she helped me out. And then Chang, another person in my lab who helped a lot with the recent data. And then funding within Villanova for a sugar in summer 2014 and a VERF in summer 2015, and then outside sources including the Research Corporation for Scientific Advancement and the Elsevier Journal/Biochemical Journal of North America. Thank you, and I'll take any questions.
Are there any questions?
Okay.
So, on behalf of the library and MERC, I am pleased to present you with your Falvey Scholar Award. Thank you so much. Congratulations.
Thank you.